How's it going, Jay? Yes. Yes.
Oh, so my friends could,
Yes.
okay, because I think at the very beginning, she emailed you and you said you didn't think you were going to. So, but now it's working out, so I can tell her to tune in.
Okay, but the plan has always been to have it zoomed.
Oh, wonder who she talked to, what?
And now we also have it on,
Okay,
what's it called?
come on, tell her, great,
Good.
okay, maybe she talked to the office. Anyway, they live in Chicago,
Yeah,
and
you can
so,
call them up right now and let them know it's,
yeah, tell her,
so they
thanks,
can watch it on YouTube also.
okay,
All
thanks.
right, here we go. You're welcome. So when you get in, have you joined the Zoom yet?
When I took embryology, we used frog eggs. Do they still do that?
They do, yeah. I'm a, team Zebrafish, but there's still a lot of Xenophis research. Is that the frog you were using? Or is it a different species?
I don't know what.
We got caught eating them. They didn't taste well.
I don't know.
Brad said that they
Is there a focus coat?
correct. Where it was gonna get easier until got into this psychology class and you're taking salivary glands out of fruit flies.
No,
And dissecting the salivary glands to get chromosomal material.
Hey, Jay, can you hear? Can you give me a thumbs up if you can hear? Thank you, Jay.
And then it got worse from there.
My internet is having some issues connecting.
No signal.
No, it says I have all the blowers.
He doesn't use a real camera.
Well, he's tried.
I think that was a presentation that came out with a screen all just like green,
Green.
this greenish light. Yeah.
No, that was the one with the, yeah, with the real camera.
Yeah, for some reason, I'm just not getting any.
Okay, can you try the other? So I'm on Edgerome.
Mhm.
Have you tried that or UWNet?
I'm on Edgy Room, and that's what's just giving me loading
Okay.
screen. Let's see. switch back over to DWNet and see what happens.
Okay.
Well, it's 1 o'clock, so I guess we're ready when you're ready.
Good. Give Christina a chance here.
So what happened?
Thanks for coming today on a cold wet one. It's good for beer and bad for Play-Doh.
Yeah.
Just made that up. So
You made that up, I
Yeah.
believe it.
nobody laughed. You're over everything. Tonight, Wednesday night at the lab, we have Allison Bennett, who's a professor in psychology, and she's also the director of the Research Animal Resource Center here. And she's going to be talking about animal research, why do it, and who benefits from it. And then next week for Wednesday night at the lab, Luke Rickert, who's a professor of history and has also got an appointment in the School of Pharmacy, is going to talk about the history of pharmacy at UW-Madison. It goes back to 1883. The pharmacy experiment station and the agricultural experiment station were established in the same year, in the same legislation, and they occupied the same building, South Hall, which is where Babcock ended up doing his Babcock milk fat test in 1890. And at that time, pharmacy was much more closely aligned with botany because so many of the drugs were compounded from plants. He's going to be talking about the new center in part on about the new center on psychoactive drugs, which we're going to have a full talk on. So those are the 2 talks coming up in the next 2 weeks at Wednesday night lab. You can also watch them by Zoom. It's a different length than this one. Or you can come to the room 1111 Genetics Biotechnology Center, 425 Henry Mall. Any luck?
Yes. Let's see. This is the only place I have the Zoom link, I believe.
So while we're, for the folks at home, we're waiting for the slow internet, which is because of the rain today. I don't know why rain screws up internet as much as it does, but it does at my house. It seems to here. Let's see. Make sure I don't do anything stupid like stop them. See what's going on.
Would you mind emailing me one more link? That's telling me that the personal link name is not valid.
Try to do that. Still working on it, and can I get your email address?
C Hansen, HANSEN 24 at wisc.edu.
Yeah, some masks you can't breathe through. I remember
Okay, did that come to you yet?
working with hepatitis patients in the hospital. God, this is a lot of work.
Yes.
Just before going into the.
Okay, so as soon as you come in, I'll make you a,
Boy, I'm glad it isn't true for everybody in the hospital.
good.
And then this came. Pandemic happened.
And now I'm gonna make you the co-host. And now you can share your screen.
Yeah, it's very important that you share.
Yes.
And I think I'll be the,
And should I mute my computer for myself?
yeah, if you can keep yourself muted, I'm gonna use this as the microphone here, I think. Hey Jay, can you come on video so I can see you and Karen too, so you can let me know if the audio is okay? Okay. Okay.
Yeah, a little bit of British sense.
All right. I
Yeah,
think
hopefully
we're,
that's not too psychedelic.
They were adults.
and if you can stay right there, that'll be great.
Sure.
And Jay and Karen, can you let me know? Can you hear us all right? Thumbs up. Jay, can you give us a thumbs up if you can hear us all right?
Yes, I'm fine.
All right, now I'm going to mute or if everybody could mute. I apologize for the delay. And thank you very much. Christina, can you tell us about yourself, the award that you got, why you're here, and all this cool stuff that you're going to do?
Absolutely. So I'm Christina Hansen, and I'm a recent PhD graduate from UW-Madison. I'm originally from Oklahoma, and I did my undergraduate degree at a small liberal arts college called Oklahoma City University. And then in 2015, moved here to Madison with my husband to come do grad school. And so I did my PhD in the lab of Dr. Francisco Pellegrini in the Department of Genetics. And I actually just finished my degree in June of this year. And so now I'm working as a postdoctoral research associate in the same lab, working on finishing up some of those projects and also chasing down leads. And I was invited based on an award I received as part of my dissertation work, where I wrote a chapter for the public. And so instead of just having the very technical, extremely scholarly and jargony work in my dissertation, I included one chapter where anyone even without a science background was able to kind of understand the work I did, and also kind of what motivated me to do it, which pretty simply for me is just because I think embryos and the beginning of life is really, really cool. You kind of have to, right? We all started as embryos. And so anyway, based on that award that I received for writing that chapter, it was posted online. And so the organizers reached out to me to see if I'd talk about it in person as well. So here I am. And what's kind of the basis of our research in the Pellegrini lab, which he's been working on for the past 2 decades, is the maternally driven part of development. So beginning at fertilization, all animal embryos, whether it's a fly or a fish or a human, you don't start out using your genome to make new products. So actually, you have this pre-packaged set that was put into the oocyte by the maternal parent of RNAs and proteins. And it's those RNAs and proteins that are delivered by the mom that drive the first few hours to days of development, depending on the species. And so here in this slide, that's what this maternal green gradient is. So we start out entirely maternally driven. There's nothing being produced by the embryo's genome itself. And then depending on what species, after a few hours or days, the zygotic genome wakes up, starts making its own products, and the maternal products are degraded. And that transition, so kind of that in-between area is called the zygotic genome activation, which makes sense. But our lab is really one of the few, I think, that focuses almost entirely on this maternal side of development. So a lot of times when you think of development, you might think of organ growth or like limb bud formation, but we're really far removed from that, like, you know, single cell to 2 cells to 4 cells, just very early processes. And one of the early processes that I focused on during all my doctoral work, now as a postdoc, is a subset of those maternally inherited products that are responsible for giving rise to the germline.
Hey, Christina, could you restart your video on Zoom? And then,
Sure.
good. It's not coming through on the Zoom. See the little start video down there?
So I stopped and restarted.
Mhm.
This. And this is what I'm seeing.
And then if you can turn that on. You're sharing.
So I stopped this video.
Yeah.
Better?
I hope so. Let's see how it looks.
Okay. Okay. So the maternal parts that I'm interested in are called germplasm, hence the title of the talk. And germplasm is just a collection of those maternally inherited products that then are carried along in the first few hours of development. And then after zygotic genome activation, the few cells that have the germplasm within them are called primordial germ cells, because then only those cells undergo that fate specification change to then eventually become sperm and egg. Whereas everything else in the embryo, they differentiate into different somatic or body cells. And so not every species does it this way. So zebrafish, along with some insects like Drosophila, and then also some amphibians like Xenopus frogs use the same germplasm method. Some mammals like humans and mice do not. So there are differences there. In the other species, they do it through what's called germline induction, where it's not anything that was pre-packed into the cell. There's actually communication between tissues later on in embryonic development. But what we learn about germplasm is still relevant for reproduction in all types of species because the genes involved are widely conserved. This one?
I apologize. Thank you.
Is that better?
It's still showing your title slide, and I don't know why. But go ahead. The folks at home can see in the little what's happening there.
Okay. So as mentioned in the title of my talk, my lab uses zebrafish as our model system for studying embryonic development. And you've probably seen zebrafish even in the pet store. They're sold widely as this glowfish or the fluorescently colored zebrafish that in the wild, they're native to India and surrounding regions. So down here, this is actually rice paddies. So they really like shallow, slow moving water. But they also adapt really well to lab conditions. So over here, this is a picture from the Zebrafish facility where I work in the genetics building on campus. And so in the middle, we kind of have breeding tables, and then on the sides, just tons and tons of tanks of zebrafish with different genetic backgrounds or porter genes where we can look at different systems. And so they're a really good model system, not just for the reasons that any animal is used for research, which might be the well-sequenced genome or lots of resources, but they're also very similar to other animals you might care about because they're vertebrates. They have a backbone, and they also share about 70% of genes with humans. So even though they're fish, there's a lot that can apply. And then for our purposes for studying early embryonic development, they're very accessible. So if you can think about in a mammal, to learn about this stage is typically happening inside of the uterus, right? So it's very invasive to get access. But in this species, they have external fertilization. So just the egg and sperm released into the water, and then you're able to very easily collect them from the bottom of the tank. And just huge numbers of embryos. You can get 100 to 300 from just a single mating pair. You can set up, tens of mating pairs at a time. So it's really easy to scale up if you need to. And they also have very rapid early development. So this shows the amount of development that happens in a single day for zebrafish. So you start out right after fertilization, where you get the first cell up at the top. Then down here, this is all yolk. So that'll provide nutrition to the developing embryo. And then by the end of the day, you have what actually starts to look like a fish, right? The head down here, and then the tail extending out. And at this size, or at this stage, it's about 2 millimeters long. And at this stage, it's about 0.5 millimeter across. So as far as eggs go, or embryos go, pretty large, which makes it easier to work with as well. So if the internet will cooperate, yeah, you can actually see the in-between. So all those cell divisions, those are actually happening every 15 minutes. So like I said, very rapid. Oh, Zoom just closed on me.
The whole thing closed down here?
Yeah. So just click me on.
Okay. And start over again, that would be great.
Oh, I'm viewing your screen actually.
Okay, so what I'm going to try to do is get me to be the screen share
Okay.
and
Well, I don't have access to that online.
okay, I'm screen sharing. How do I make this thing big?
Here you go.
There we go.
Okay.
Up, so you can see the tissue coming down. And then up here, you'll eventually see an eye start to pop up.
Yep.
Yep, there's the eye.
Mhm.
Yeah. So it's really neat. Been a really good system for studying those early stages of development. Am I okay to continue?
Yeah. Karen and Jay will have to watch the little bit that's coming through here, and I'll try to figure out how to get it.
Okay. I can also send you my slides later if they want a
Thank
hard
you.
copy. So thinking back on that video I just showed you, those early first cleavages where you can see it going from the 1 to the 2 to the 4 to the 8 cell stage, that's when germplasm is being aggregated in between those cells. So really in these first 3 to 5 hours of development. And then by the 6th cell stage, they're called primordial germ cells officially. And those primordial germ cells start migrating to what will eventually be the gonad. And then as the gonad develops, the eventual either sperm or eggs will mature, and then the whole reproductive cycle starts all over again. So here I'm going to go into more detail about what germplasm actually looks like during those first few stages. And so several labs, including our own, have actually observed that germplasm is what's called a biomolecular condensate, or a phase-separated structure. So what that means is that rather than being contained within a membrane, like the nucleus or mitochondria, it's just in a different phase. So think of like oil and water, where they don't mix, even though there's no real physical barrier between them. And so it behaves almost like liquid droplets within the cytoplasm. And so here you can see that in this one cell stage embryo, that these germplasm particles are just dispersed. And then they actually compact into these much larger aggregates. And they form 4 large germplasm masses at the end of what we're calling the cleavage furrows here. So the site where the cells are dividing at the very end. And then those 4 aggregates continue to compact together, and they're divided asymmetrically into cells as development progresses. So that means, you know, instead of like breaking in half and going into 2 daughter cells every time there's division, for the most part only segregates into a single daughter cell. So you maintain only a population of about 4 cells that have germplasm inside of it.
There is no nucleus now?
There is a nucleus.
Okay.
Yeah, oops. So if you look in this image here, it's right next to the nucleus in blue. And so it's larger than the nucleus, which is, you know, kind of tells you how crazy huge the structure is. And in green, we're labeling cell boundaries for these last 2. So now you have many, many cells. Each one of this is a nucleus, cell boundary, but then only the cells that had germplasm in them, that's now dispersing into those cells, and that's what's going to give rise to the germline. And so in each of these, I've just labeled a single germplasm component. In purple, that's the RNA called Nanos, and it's highly conserved in the reproduction of all animal species, you know, human to fish. And it's, you know, a very important component of zebrafish germplasm, but it's, you know, one of at least 12 RNAs that we know of. And we also know of 4 to 5 proteins that are associated with the germplasm. And so it's really interesting because you have all these different products that have to compact together and then stay relatively stable, even though they're going through so much, you know, perturbation within the embryo. So you saw it really moving and dividing, and it's getting just transmitted from cell to cell in an inactive state, we believe, until after it actually hits this final stage where it spreads out into the entire cell. And we also know that if you remove the germplasm, so either at these early stages, if you suction out those masses, or later on, if you just selectively kill the cells that have it, the adult will be sterile. So they're absolutely essential for reproduction. But for such a critical process, not much was really known at the molecular level about how it's structured, especially in a vertebrate like zebrafish. So for the first part of my talk, I'm going to really focus on how this germplasm is structured in general, using these 4 RNAs. So if you see these names a lot, those are just different RNA components of germplasm that I'm using as labels. And then I'll also use a germplasm protein called Buckyball or Buck for some of my labeling. And unfortunately, it wasn't named for UW-Madison. It was like the toy Buckyballs, little metal balls.
Who's it named for?
I think that it was named in Germany, but it was for the ball toy. like buckyballs.
My understanding was for Buckminster Fuller.
Oh, was, yeah,
Yeah,
like the Buckminster lattice.
not sure, but
He did some of the geodesic domes.
Gotcha. So that structure?
Yeah.
Interesting. So here you're looking at top-down view of a 4-cell zebrafish embryo. So each of these corners is a cell. And then at the end here, all of the germplasm masses after they've been aggregated. But even after they've been aggregated, there's still these unaggregated particles just scattered around. And so I was interested in the structure of what they're like when they're at this particle stage. And so here's an example of my results when I label 2 different RNA components, in this case, VASA in green and dead end in magenta. And in this image, what I want you to notice is that even though the particles are often very closely associated, they don't really seem to overlap within the same particle. So another way to get a view of this is if you zoom in on one of these little particles, and we've actually estimated that, so one of these bright dots would have around 9 individual RNA transcripts inside of it. And so you're not looking at just a single RNA, you're actually looking at a cluster of similar RNAs. And even though they look very close together, we image these in a way that we can represent them in 3D, right? So it's not just like a snapshot on your phone, it's going down in the Z dimension. So you're taking basically slices through a cube, if you can imagine it that way. And so this is a montage of every single slice through that cube going down. And so you can see even this is a little deceiving because there's actually quite a bit of space in that dimension between the green and the magenta, which in this case are 2 different types of RNAs. And I did this type of pairwise comparison between at least 6 different RNAs and every kind of pair you can imagine. And it was always this pattern of homotypic or non-mixing, which was really surprising for us because for something that's phase separated, you'd imagine it would act more like a liquid and maybe fuse together. But we really didn't see any type of fusion. And so that also got us thinking, like, well, how do they know to discriminate based on type, right? Like how do the dead end transcripts stay with other dead end, and how do the VASAs stay with other VASAs? And there's nothing in their sequence itself that gives us really clues. So that's kind of a big mystery for us still to see how they're able to do that. Maybe there's some unknown protein that helps keep them apart. But then even more surprising is that even after they end up very tightly packed into those large aggregates, they maintain their homotypic or non-mixed nature. So for this, you can see, yeah, those particles still have their distinct identity rather than being forced to kind of fuse. And to measure this, I just used Pearson's correlation coefficient. So where 1 would have been perfect, like colocalization in the same spot, and instead you're down at 0.32. And even that's an overestimate because that was just with these 2D flattened projections rather than that 3D, slice by slice in the cube. And we also did control experiments to make sure that it wasn't just some artifact of the imaging, like maybe one laser is slightly off from the other laser. But when I do the same RNA labeled with 2 different colored probes, so you would expect these to overlap completely. You see that, it's very different from when we label 2 different ones. It's right on top of each other. So something else I wanted you to notice from this slide really was the branching off point of some interesting work we've done. Just because this very weird structure. So rather than just like a rod, we kept seeing that germplasm aggregates had the spiral appearance. And it was very repeatable. And this is a new finding for us and the field because traditionally, and by traditionally I mean like early 2000s, this is kind of the best you got when you image germplasm. And so, you know, it was a blob, sometimes irregular, but you couldn't tell much about its structure. And I think no one was really expecting, now with the higher resolution and better microscopy techniques, that it makes this very interesting structure. And with the imaging I do, we can make 3D reconstructions. So this is showing the same one from a different angle. And it's definitely, when you rotate it around, it's definitely this weird helical structure. And so my boss and I, really thought about it, and we're like, okay, well, why would the embryo do that? Maybe it's beneficial in some way. And so we were trying to get inspiration from nature, because there's many other helices in nature, from the very famous double helix of DNA to even just like a climbing plant tendril here, or shells, or those horns on the mountain goat. A lot of times helices can be really useful for just structural integrity, especially when it comes to sphere packing. And so some of the reading I've done says that that's the most efficient way to pack a bunch of spheres into a cylindrical space, is if it forms this helical conformation. And then some other things we thought about was maybe that's useful for the contacts between different molecules. Maybe depending where it is in that large macrostructure, it might be, one molecule might be in closer proximity to something it might be active with later. And so those are some big questions we're still trying to answer. But that's at least where our head's been at, as far as why would this matter? Or is it just a fluke of, energetic, like biophysical associations between the molecules? But one thing we kept running into was that the structures are very variable. So it's not, a single, like always left-handed regular helix, or always right-handed helix, or anything like that. They're very variable. And so we wanted to have a better way to characterize the structure. And also it's hard to publish a paper just saying, look, it looks funny, So I worked together with 2 scientists to help come up with an image analysis system to better characterize those shapes. And so in brief, we take the images, and so every single slice I've taken through that cube, so we get the full 3D representation. And for each of those slices of the cube, detect each point here, so based on just isolating regions of fluorescence from the background. And that gives us an output with like X, Y, and Z coordinates, so we can map it in 3D space. And then once we have that, map on a computer, then you're able to do like curve fitting algorithms to determine, what actually is the equation of a specific aggregate. And then also measure some parameters like pitch, which is the distance between curves in a helix, and then also things just like radius through the cross section of the helix. And then ideally take that and compare from different conditions, different developmental stages. And we are still actively working on this. We have it up and running, but it's an average of the entire aggregate rather than just one part. Then what we'd like to do is a moving window, is basically what I'm trying to say here. Because when you average out the parameters, turns out you lose a lot of information, especially when you have one end of these structures that might be very tight and spirally, whereas the other end becomes more flat. And so that's something we're still working on quite a bit. But we've made some exciting progress in this area. And so moving beyond that, something I was immediately interested in was whether or not the cytoskeleton is actually responsible or required for forming this helical structure. Because if you notice earlier, they only form at the furrow ends of the dividing embryo. And so this is a cross section looking down all the way across the cleavage furrow. So that's where two cells are going to divide from each other. And so at either end, you have germplasm, and then magenta here, you have microtubules. So you can see they're really closely associated with microtubules. And then you can see it again here, higher magnification, just right nestled in there. Then also imaging germplasm with actin, another cytoskeletal component. It goes along what we call the actin ridges that span the outside of the embryo. And so, I thought it was reasonable that maybe it's the presence of those cytoskeletal elements that helps promote that helical structure. And so to test that, I took advantage of a mutant we had in the lab called Rna, which has defective furrows and, importantly, really defective cytoskeletal organization. And so we already knew that these Rna mutants can't get the large germplasm aggregate. So this would be in a normal, not defective embryo. But then over here in the mutant, you still get clumps, but they're much smaller. But even though they're smaller, I figured I could still image them to see if there's any type of structure going on. And sure enough, there was. So these are all images taken from those Rna mutants with the disorganized cytoskeleton. And every single one of these aggregates is found away from a furrow, so not nestled into those cytoskeletal elements. And they're about 1/10 of the length of normal germplasm, but even so, they still form this, spirally curvy helix-like structure. And so that told us that there's something, intrinsic to these molecules themselves that promotes this type of formation. And then something else we were wondering is what stabilizes those large germplasm aggregates? Because like you saw really well in the Rna mutant, even in the normal ones, you'll get little bitty aggregation going on outside of the furrows, but then they're normally degraded before that's able to make primordial germ cells. And so it seems like there's something about a certain threshold for it needing to be a large one for it to actually do its job in promoting germ cell fate later on. And so far, I've really been focusing on the RNA components, but now I'm going to talk about that Buckyball protein. And so Buck was the first one identified as essential for germplasm organization in zebrafish. And this just shows its localization. So the really classic in the furrows of zebrafish embryo. And no one had imaged Buck protein with germplasm RNAs. And there is no obvious RNA binding domain on the protein itself. So it was really unclear what type of interaction was happening between these molecules. And my data showed that it seems like a subset of RNAs do associate real closely with that Buck, whereas others, like down here, there's more separation. But then when it's actually aggregated together, so now Buck is shown in this yellow green and two different germplasm RNAs, dead end. In the first panel, dazzle, and the second panel are in magenta. And what we saw, and hopefully you can see here, is that it almost appeared that Buck is forming this kind of scaffolding core where the RNAs are either wrapping around it or just decorating the periphery. And so that, for us, kind of led us to this very simplified model where there's this Buck protein core that's helping to stabilize the RNA particles wrapping around it. And we're starting to get evidence now that it's not just Buck, but there's maybe a few other proteins that are involved in forming the stabilizing core. So far I've told you about the structure of germplasm from those individual particles to the aggregate. But for the rest of my talk, just want to briefly tell you about another project, but this time instead of just zebrafish, it's multiple fish species. I think it's pretty fun. So traditionally, primordial germ cell research has occurred in very distantly related animal species, kind of the outcome of having, the certain set of the traditional model organisms, right? So really just C. elegans, or the nematode worms, Drosophila flies, zebrafish, and then xenophus frogs are really the bulk of this PGC research that exists in the literature. And some of it can be very informative because there's some characteristics that are shared, like the fact that it's phase separated, that's in all these species. And then some of the genes like Nanos and Dazzle are conserved. But there's also some important differences between germplasm and these species. And we were curious to see what kind of differences or similarities would occur when we look at much more closely related species. And so a few years ago in our lab, so my boss was really inspired by work other labs had done on actually pigmentation genes, because you could see how, this would be really rife for pigmentation research. But he thought we could get a bunch of different related species and start doing evolutionary developmental biology research in more reproductive area. And so now we have nearly all of these species in the lab, and a few that aren't pictured as well, in like little research colonies. And I decided to start my comparison with just 4, thought it was a reasonable number, and that includes zebrafish. And the 4 I picked were at different, places on the evolutionary tree of this family of fish. And so starting with zebrafish at the bottom, a very close relative, diverging for less than 7 million years estimated, and then slightly more distantly related. And then the sister genus, so no longer a Danio, but still relatively closely related. And just to put this in context, because it's, it can be weird thinking about evolutionary distance, especially in fish. So there's another fish that's had a fairly decent amount of germplasm research called Medaka up at the top, or Japanese rice fish. But that's been diverging from zebrafish for approximately 150 million years, which if you go back to this, humans and mice have been diverging for just 90 million years. So that tells you really how distantly related and how many changes could have occurred between the genes of these 2 fish species. And so, yeah, we're really zooming in on closely, in relative terms, related fish species. And so first, I just wanted to see if, germplasm was there, or if we were able to label it. And so I used Nanos Rna Probes, since Nanos is one of the most highly conserved reproduction genes. And these labeled probes that I used were actually the ones I had designed originally for zebrafish, because none of these other 3 species had a sequenced genome, so I couldn't just take the sequence and design something for them specifically. But I was banking on, that hopefully Nanos was so well conserved and that they're so closely related that it would cross hybridize. And luckily enough, it did. So you can see in all of these 3 non-zebrafish species, you get that germplasm aggregation in the 4-cell stage embryo. And then, so once I knew that it worked, I was able to go higher magnification and at a few key stages of germplasm aggregation. So in this first column here, this is again at the 4-cell stage, but really zoomed in. And you can see that helical or helix-like structure is conserved between all these species as well. And then this is part of the segregation phase of germplasm behavior. So this is when it's going to just a single daughter cell. And so now the cell boundaries are in that magenta. You're looking at a nucleus, each of these in blue. and then Nanos labeled germplasm in green. Then I also went to what would be the dispersal phase, which in zebrafish is about 4.3 hours post-fertilization, where that germplasm goes back into discrete particles that fill the resident cell. And you can see a nearly equivalent process happening in all of these other species as well. And so after I established those very similar patterns of germplasm localization using nanos, I decided to test every single known RNA component of germplasm in zebrafish to see if it was also present in all these other species. And this is just the list. You don't, really need to focus too much on it, other than just to say that these 5 RNA components have never been identified in the germplasm of any species other than zebrafish. So I was really curious to see if any of these were also in one of these related fish species, or if there is, some species-specific components. I was thinking they'd most likely be there just because, genes involved in reproduction are typically very conserved, but certainly curious to test it. And so here is the final product after many embryos. And as you can see partially, and I'll also zoom in on a single panel, for nearly all of those RNAs, they were in fact present in the germplasm of all the other species I tested. And so this is looking at hook 2, which is actually the most recently discovered component of germplasm in zebrafish. I believe it was in 2018 that it was just discovered, but it was in all of the species. But there was one exception, and that single exception is an RNA called CA15B, and it was present in all of the Danio species, but it was absent in the germplasm of Divario equipinatus, which is also sold in the aquarium trade as giant Danios. It is actually a different genus than Danio. And so to see that a little bit better here, so here's a 4-cell zebrafish embryo, and the zoomed in insets where you can see CA15B, that germplasm localization in the furrows, but then in the Divario equipinatus, not only is the embryo itself just brighter in general, you don't see any type of specific aggregation or germplasm signature from that CA15B. So looking at this, I thought that it was likely that CA15B RNA is present in the embryo because of how bright it is. I was thinking, maybe that probe is binding something, but it just happens to be distributed all over instead of specifically going to the germplasm. But it also could have been a fluorescent background, so I tested that using reverse transcription PCR. So I used that to determine which RNAs were being expressed at certain stages. And so I made sure to focus on 2 embryonic stages that would be during the entirely maternal phases of development. And so these 2 bands here highlighted in yellow, that's showing that CA15B was indeed present in the embryo at the 2-cell stage and at the 1,000-cell stage. And so that tells me that CA15B is being expressed, it's maternally inherited, so it was packaged into the egg, but for some reason in this species, it's not going into the germplasm. And so that led me, thinking about what effect, if any, the lack of germplasm localized CA15B might have on the primordial germ cells of this organism. So CA15B encodes a protein called carbonic anhydrase 15B, and it catalyzes a reaction that affects pH and cell polarity. And zebrafish, it's actually responsible, or partially responsible, for the migration phase. And so taking those primordial germ cells and giving them the polarity cues to know which way to migrate to end up in the gonad. And so that, is part of what I'm picking up on now as a postdoctoral researcher is figuring out how these Divario fish accomplish the same process, because clearly they're able to build a gonad or they wouldn't ever reproduce, but without this player. And so I'm trying to figure out if, very redundant function is being played by a different gene, or if they have a different form of migration entirely. So that's where I will wrap up this second story. I'm very excited to keep going on, especially with this CA15B. Maybe I'll be able to update you in the future. But thank you everyone for your attention, especially with all the technical difficulties. And I'd like to thank my lab, have some wonderful lab mates. This is Francisco Pellegrini, my boss, some of our dogs. And had a lot of really helpful collaborators, both at UW and others in the Zebrafish community. And these were the members of my PhD thesis committee. So with that.
Is your boss the new department chair?
He is, yes.
Okay. How does that work out for you?
For me?
Yeah.
Oh, he's very busy, but it is nice because I think he now has more reason to talk to kind of everyone in the department. So if I'm talking about a technique we don't have experience with, he'll be like, oh, so-and-so, I think is working on that. You can check with them. So, but there's definitely pros and cons. He is a lot busier than he used to be.
And are all the genetics faculty in the building, or does he gotta go to places?
Oh yeah, he's got to go to places. We have most of the genetics faculty in the building, but then he is also, so he's technically the chair of the Laboratory of Genetics, which includes the Department of Genetics, which is in the College of Agricultural and Life Sciences. But he's also the chair of Medical Genetics, which is in the School of Medicine and Public Health. So he has two deans to answer to, two large colleges, and that's mostly, at the hospital campus.
Got a very practical question.
Yeah.
Got all these tiny little fish and all these aquariums. How do you keep track of them? I mean, you can't put hello, my name is sticker on them.
Right. So, we have to be very careful about labeling the tanks. And within a tank, it'll all be siblings, so from the same parental cross. And so they're at least the same, genetic background. They're not as inbred as you might hear about. There's very inbred strains of mice where you really know that they have extremely similar backgrounds. We try to keep a little more genetic diversity just to keep them as healthy as possible. But we segregate them by background. And then if we need a specific zebrafish, we have a section where we put them in just a little individual tank. Yeah, but they don't like that. They don't do as well because they're very social fish. They love schooling in their natural environment. So they're much happier if there's at least 10 of them in a tank.
There's no way you can verify after the fact that you take them out to the tank.
Right. No, we don't do any type of DNA barcoding, for instance. That's something another researcher in the lab is looking into, but for conservation purposes. And so using our fish is a way to kind of work out some optimized way of doing DNA barcoding, but we currently don't do that. In the back?
The germplasm, splitting off into 4 places, and then maybe splits off even more, but then it comes together as it becomes a gonad. Is there any understanding as to why it does that, as opposed to just staying in one place?
Right. What we all kind of guess, but haven't tested empirically, is that it's just a way of ensuring that there will be enough germ cells. It's a difficult balance because we're not entirely sure that every single germ cell or primordial germ cell that has germplasm in it necessarily makes it all the way on its journey to the gonad. We haven't done live tracking to see that, but a lot of early development is characterized by robustness and redundancy. So we think that might be one form of redundancy, is just making sure there's enough progenitors that do make it to the gonad.
Yeah, when I was looking at those great images of the helical structure there, it kind of looks like maybe the different types of RNA are not uniformly distributed.
Absolutely.
And I was wondering if the variations in that distribution could maybe account for like the pitch and the radius of the helix.
So that's something, yeah, that we're very interested in. So I didn't mention it in the background here, but there's actually 2 starting pools of RNA in the egg itself. There's some that start up in what's called the animal pole, which is closer to where that first cell forms. And then there's just one RNA called dazzle that's, well, actually there's 2, dazzle and self1, that start at the vegetable pole, which is down at the very bottom of the yolk. And those 2 have to travel all the way up to join the germplasm. And it seems like those 2 particularly are very differently distributed when they enter the germplasm. And so we're trying to figure out, you know, A, if they impact the pitch, like you said, like maybe when they come in, they help compact the structure, make it denser and more stable. And then 2, if there's something about the structure that, you know, promotes a certain ratio, like maybe you need a fourth of it to be all dazzle and the rest can be a mix of the others, or, you know, something like that. So now we're trying to think of ways to, you know, maybe overexpress a certain RNA or deplete a certain RNA and see what happens to the structure and to the germ cells later on. But, you know, that's exactly along the lines we're thinking, because we noticed the same thing. It's the distribution can be really different depending on the RNA.
A less related question, but I'm thinking of getting fish. Do you recommend getting zebrafish?
Oh yeah. Yes, zebrafish are great. Like I said, they really prefer to be in groups. So as long as you have a big enough tank to where you could accommodate, I think 6 is the bare minimum, but they're really happier if you have 10 to a dozen. And it also depends if you want a community tank with other species, because they're very active. And so if you have like much slower, easygoing fish, I know they can start to kind of terrorize them, not because they're mean, but just because they're constantly swimming around. But they're great fish for at home. So yeah, I highly recommend.
I know for a lot of the mammals that we hear about that are in experiments, I know that, you know, these ethics boards and stuff, you have to.
Yeah.
This is a similar thing for fish. Do you have to go through? Okay.
There are, yeah. So some of it's a little bit different. So fish aren't considered vertebrates in a legal sense until that backbone actually starts being formed and the nervous system starts setting up. So like when we collect the very early embryos, they're treated like invertebrates. So there's not all those governing boards for like microinjection and stuff. But once they're older than about, I think it's 3 to 5 days past fertilization is when you start having to think about, you know, like humane euthanasia, for example. There's protocols for euthanasia. We get monthly vet visits to make sure our facilities are well kept. And so there's pretty stringent ethics as far as like animal welfare. Not as much as far as embryonic type research early on, since they are so, I guess, underdeveloped at that point.
Does that hold for other species if you're working on mice or rats?
I think mice and rats have a lot more governance, but I don't know many of the details. I
Okay.
think it's kind of rare that, so we manage our own fish facility. We don't have any like outside staff coming in, whereas I think mice and rats facilities often have kind of an external
Okay.
like staff or husbandry staff.
So I'm not sure, but I think mice and rats are under USDA. Are fish under FDA or Fish and Wildlife or somebody else?
The only board I'm aware of is the IACUT, which is the institutional
That's-
animal care
yeah.
use. And that's here. I'm not sure about federal agencies.
Yeah, me neither.
We're funded by the NIH, so anything the NIH rules, we have to comply with, of course, but that's the only federal agency I'm aware of.
I have two questions. The
Sure.
first is, how long do zebrafish live?
Yeah,
Is that a complicated factor? The second question is, are there researchers doing work on germplasm in any simpler organisms in zebrafish?
yeah. So to answer your first question, in the lab setting, so this goes along with some of the humane euthanasia and husbandry protocols. So we're supposed to only let them live for about 2 years because that's really the extent of their very healthy and like good quality of life years. And so after 2 years of age, except in extenuating circumstances where we have like a very special mutant that we can't get to breed or something like that, you're supposed to go ahead and start the next generation and make sure you have younger ones coming up, and then humanely euthanize the
How do you do that?
ice. Yeah, so you typically you net them and then you put them in a bucket full of very like icy cold water, which slows down their metabolism, basically puts them in like a sleep light state. And then once they're completely stopped moving and relaxed, then they're frozen. Yeah.
There's enough room on the door for both of them. It's Titanic.
Oh no.
Sorry.
But I mean, yeah, in very cold water, everyone, shuts down. And there's been a lot of work on it, and it seems like that's the way that stresses them out the least, because it used to use different drugs where they'd kind of panic, but the slow cold seems to be the best.
And then you don't have to cut their heads off or anything?
No,
You
just deep freeze.
In some human cultures, they use ice.
Really?
Well, that's a terrorist group.
Oh, well, that's not great.
I thought I made a
This is
bad
the second
joke.
question.
There you go.
Second question, yes.
Will you remind me what that is?
Are there researchers doing similar work with germplasm in simpler organisms?
Oh, sure, yes. So there's been a lot of germplasm research and fruit flies specifically. So they are a little bit different because they don't have those furrowing, to make the cell divisions. It's more of an interesting, I'm not very familiar with flies, but they don't have those furrows, and so they don't get those 4 aggregates. But they do have those very dispersed particles, like the very first stage I showed you, where it's the liquid-like germplasm granules. And then they all go to one side of the embryo. And so they also segregate in that manner just by going to a single side.
So you answered a question about, I think it was Paul said, was there a nuclear membrane the whole time? My memory is there's folks that go through cell division, and part of that is the nuclear membrane goes away and then reforms.
Oh, yeah.
Do you have any examples of embryogenesis with stuff like that, where the nuclear membrane goes away and then reforms, or?
So during like the actual cell division mitotic process itself, it would go away.
Yeah.
Yeah.
But does that happen with your folks, or do you have a?
Oh, it does, yes.
Oh,
Sorry.
okay.
Sorry, I misunderstood. I thought nucleus in general. I didn't hear the nuclear membrane part of the question earlier. But yeah, the nuclear membrane does get broken down and then remodeled during mitosis during these stages.
These guys, okay.
Yeah, but actually really early on. So the first, I think it's 3 hours of zebrafish development. There's no checkpoints on mitosis. So that's how you get those very rapid, every 15 minutes of cell division. So it's just, the action part of mitosis where you have the chromosomes condensing, separating back together. There's no checkpoints or gap phases.
And in plants, a lot of times you get multicellular, one cell, but lots of nuclear, I should say.
Right.
Is that what's happening here, is that you get a lot of mitosis without cytokinesis?
You don't get complete cytokinesis. You just get a partial furrowing until about the 32 cell stage. And so early on, where germplasm is actually existing is kind of an in-between. So it's in the furrow, but there's no firm boundary between cells. And so it's, you know, very leaky and porous between those first cells.
And if you do disruption experiments, what's the time course of taking 1 or 2 or 3 of those out? When does that start screwing things up, or does it never screw things up? They have enough orientation that they
Of
can.
the cells themselves or the germplasm?
So you got 16 or 32 cells.
Right.
How many of those can you take out at that point without
Oh, okay.
screwing up the further development?
Ooh, I'm not sure. Yeah, I honestly don't know. Yeah, I can tell you. That's interesting.
That's one of the cool experiments about how you can make identical twins just by cutting it.
Right,
People were doing that like 40 years ago,
right.
at least. And that was always pretty fascinating to me, is how far could you get before the poles are set up
Exactly.
and then you're not getting too equal.
I would guess maybe slightly before that zygotic genome activation. So assuming you have enough of those maternal products kind of evenly distributed,
Yeah.
you should be able to divide quite a bit. And I think once the zygotic genome starts producing, then it might have some more body plan set up.
And if these fish make monozygotic twins or anything like that, or is that, don't you know?
Not that I know, no.
Any other questions? Okay, let's give her a hand. Well, if anything in the next couple of years and want to come back, you just let us know.
Absolutely, thank you.
And next week we go to algae in the lakes.
Yeah.
I don't know how the algae are with your fish, but
Boy.
Am I okay to leave the?
Yes, thank you very much.
All those blue green.